This lesson's about active transport, and here are your objectives. Now first, recall that active transport, as we described in a previous lesson, is transport across a membrane that requires energy input. It doesn't go on its own. And that energy input in cells is usually provided by adenosine triphosphate, abbreviated as ATP, adenosine triphosphate. And adenosine triphosphate is shown here in this diagram on the right-hand side. It's a nucleoside that's a ribose sugar with a nitrogenous base connected to 3 phosphates. So it's a nucleoside triphosphate. And in fact, it's actually used to make RNA, ribonucleic acid. But an important function that it also has in cells is acting as a kind of energy currency or temporary energy storage. And that's because breaking this bond, so breaking the bond between the second and third phosphate, when you combine it with water, releases energy. The reaction of ATP and water, in other words, the hydrolysis of ATP, to ADP, adenosine diphosphate, again we're cutting here, and splitting off that last phosphate, which we write like this, P sub I for inorganic phosphate, that releases energy. And because the hydrolysis of ATP releases energy, ATP itself can act as a kind of energy storage for the cell. So ATP provides the energy for active transport. Again, recall that active transport moves substances against their concentration gradient. In other words, it moves them from low concentration to high concentration across the cell membrane. Let's look at a specific example, the sodium-potassium pump. So here's a phospholipid bilayer, and embedded in it I have an integral protein. That's my sodium-potassium pump. And at the bottom here I'm going to have the inside of the cell, and out here this is the outside of the cell. Remember that sodium and potassium, because they are charged ions, cannot pass straight through the phospholipid bilayer. Again, they're ions, they can't dissolve in the hydrophobic interior. So in order to move in and out, they'd have to move through some sort of protein. Now, this protein, the sodium-potassium pump, is actually going to move 3 sodiums out of the cell, and it's going to move 2 potassiums inside the cell. And in both cases, that's against their concentration gradients, because sodium is in high concentration outside the cell and in low concentration inside the cell. In contrast, potassium is in high concentration inside the cell and in lower concentration outside the cell. So left to its own devices, sodium would be moving in and potassium would be moving out, but the pump is doing the opposite. It's moving both of them against their concentration gradient. Now, in order to do that, again, energy is required and is provided by ATP. The way that's often shown is this. ATP is hydrolyzed to ADP, adenosine diphosphate, and that inorganic phosphate we mentioned. The water is often not shown here, but it's implied. Remember that ATP is hydrolyzed to give ADP and phosphate, and that releases energy. This kind of curved arrow is showing that it's involved in the reaction, and that's happening at the sodium-potassium pump. So here we see 3 sodiums moving out, 2 potassiums moving in, and ATP being hydrolyzed. Now you might have noticed that we're moving 3 positive ions out and only 2 positive ions back in. So in fact, in addition to moving ions, this pumping action is making the inside of the cell slightly negative compared to the outside of the cell, because fewer positive ions are being moved in than are being moved out. And so if sodium were allowed the chance to flow back in, for example through an ion channel, there'd be 2 forces driving it. Both the fact that it has higher concentration outside and lower concentration inside, so it would want to move back in for that reason, and also because it's a positive ion and the inside is slightly negative, it would want to move back in for that reason as well. And so instead of just talking about a concentration gradient for sodium, we can actually refer to an electrochemical gradient. The chemical part referring to just the concentration concentration gradient, but the electro part referring to the fact that there's also this additional factor of a positive ion wanting to move into a slightly negative environment. So we will encounter this term again when we talk about nerve impulses, because the sodium-potassium pump and this electrochemical gradient are key for how impulses are sent along nerve cells. All right, let's talk about endo and exocytosis. Endo and exocytosis are forms of bulk transport, and they both require energy input. So they are forms of active transport. Endocytosis can be divided into further subforms: phagocytosis, pinocytosis, and receptor-mediated endocytosis. So first, phagocytosis, which means cell eating, is when a cell extends, these are called pseudopods, extensions of the cell membrane out around something that it's engulfing. So it can engulf a large particle. So this is the cell down here, and it sends out these extensions, these pseudopods, and it engulfs something. And something like a white blood cell might do this, for example. Pinocytosis means cell drinking, and it's a way that smaller molecules can be taken in. And what we see there is just the cell membrane, and let's say there are some particles outside of it, and the cell membrane starts to pinch off like this. And by doing that, it ultimately forms a vesicle with those particles in them. And that vesicle ends up inside the cell, containing the molecules outside. So that's pinocytosis. Smaller particles, smaller amounts of particles. Now this, as I've drawn it, is non-specific. It's just taking whatever is outside into this vesicle here. But it can be made more specific if there are receptors on the cell that are recognizing particular things and triggering this action. So this that I've drawn here in green is a receptor. It's detecting one of these particles, and it's signaling to the cell to start this pinching off process. And therefore the cell has some control over what it's taking in. It's still taking a bit of a gulp of everything that's outside, but the process has been triggered by recognizing that among the things outside, there's the thing that it wants. So that's receptor-mediated endocytosis. That's kind of a variation on this. Exocytosis is the reverse process. Exocytosis, you can think of running this picture backwards. You've got a vesicle containing some materials in it, and that vesicle moves towards the cell membrane, fuses with the cell membrane, and releases its contents into the space outside the cell. And an example of this is how neurotransmitters are released from the end of nerve cells so that they can go and trigger an impulse in the next nerve cell. So exocytosis is running in the reverse direction from what we saw up here. So that's active transport, the sodium-potassium pump, and endo and exocytosis.